Good evening and welcome to Dreamy Academy. Tonight, we begin a journey into the quiet wonders of biology, exploring concepts often misunderstood, not because they are complex, but because their beauty often hides in plain sight, waiting for a moment of gentle contemplation. As we begin, I'd love to know where you're watching from, your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. Tonight, let us begin by contemplating something remarkably intimate and yet utterly universal. The very air you breathe. Each inhalation draws in a tapestry of invisible gases, a mixture refined over billions of years of planetary history. But within that breath, something more profound stirs. The oxygen molecules you are now welcoming into your lungs have been on an extraordinary journey. Some of them perhaps once danced through the leaves of a primeval fern, releasing their bond with carbon in the sunlit air of the Carboniferous period. Others might have been exhaled by a towering brontosaurus, its colossal lungs filling and emptying in slow, rhythmic sighs across ancient plains. The nitrogen, too, whispers tales of deep time, having cycled through countless organisms, from the smallest soil bacteria to the mightiest whales, before returning to the atmosphere ready to be drawn in once more. This isn't merely a poetic thought, it is a fundamental truth of our existence. The atoms that compose your body, the very essence of your being, are not new. They are ancient, perpetually recycled, borrowed from the vast cosmic reservoir that has fueled planets and stars for eons. Every atom of carbon, oxygen, hydrogen, and nitrogen within you has a story that stretches back not just through the history of Earth, but through the fiery hearts of distant, long-dead stars. We are quite literally reassembled stardust, animated by the same forces that sculpt galaxies, breathing atoms that have known the breath of dinosaurs. This profound interconnectedness, this ceaseless dance of matter and energy across scales of time and space that dwarf human comprehension, is what makes biology so endlessly beautiful. It rewards patient contemplation, inviting us to see ourselves not as separate observers, but as integral threads in the universe's grand living tapestry. Have you ever considered what truly separates a stone from a living cell? It seems a simple question, almost self-evident in its answer. A stone is inert, cold, unmoving, while a cell divides, grows, consumes, and responds. Yet, when we begin to gently peel back the layers of our intuition, the boundary between the living and the non-living becomes beautifully, almost impossibly subtle. It's a concept often taken for granted, but one that holds the key to understanding everything from the simplest bacterium to the intricate symphony of your own thoughts. Let's begin by considering the most fundamental characteristics we assign to life, those properties that seem to whisper, "Here, something is alive." We might point to organization, the intricate, non-random arrangement of parts. A crystal of salt, though orderly, lacks the layered complexity of even the simplest virus. Then there is metabolism, the active process of acquiring and transforming energy, breaking down molecules and building new ones. A river flows, but it does not metabolize the energy that moves it. Life also exhibits homeostasis. the remarkable ability to maintain a stable internal environment despite external fluctuations. Your body, for instance, tirelessly regulates its temperature, blood sugar, and pH, a delicate balancing act that ensures survival. Crucially, living things respond to stimuli. A plant turns its leaves toward the sun. A bacterium swims away from a toxin. They also grow and develop, following a genetic blueprint to increase in size and complexity. And perhaps most famously, life reproduces, creating offspring that carry forward the species' lineage. Finally, and this is the grand orchestrator of all the others, living organisms undergo evolution, adapting over generations to the ever-changing tapestry of their environment. These characteristics, taken together, form a coherent picture, a set of rules that seem to define the game of life. But like all elegant rules, their beauty lies in their nuanced application. The deeper we delve, the more we realize that these characteristics are not merely isolated traits, but rather the visible manifestations of an underlying ceaseless molecular dance. Imagine, if you will, the interior of a single cell, not as a static bag of chemicals, but as a bustling city at twilight. Within its boundary, millions upon millions of molecular machines are at work, each performing a specific task with breathtaking precision. Proteins fold into intricate three-dimensional shapes, ready to act as enzymes, catalysts that accelerate chemical reactions by factors of trillions. Other proteins form structural scaffolding, giving the cell its shape and allowing it to move. Still others act as tiny pumps, moving ions and molecules across membranes, maintaining that delicate internal balance we call homeostasis. At the heart of this molecular metropolis lies the information itself, DNA and RNA. These nucleic acids are not just inert storage devices. They are the very language of life, encoding the instructions for building all the proteins, for regulating all the processes, for orchestrating the entire cellular symphony. This information is meticulously copied and passed down through generations, allowing life to reproduce with astonishing fidelity, yet with just enough variation to allow for the slow, patient sculpting of evolution. The beauty of this system is its self-referential nature. The information builds the machines, and the machines process and replicate the information. It's a closed loop of exquisite complexity, powered by the constant flow of energy. Consider, for a moment, the sheer scale of this activity. In a single second, within a single human cell, countless chemical reactions are taking place. Molecules are being synthesized, broken down, and transported across membranes. Genetic information is being read, transcribed, and translated. This is not a slow, deliberate process. It is a furious yet perfectly orchestrated ballet of molecular interactions. And every one of these interactions, every twist of a protein, every bond formed or broken, is governed by the same fundamental laws of physics and chemistry that apply to a stone, to a star, to the empty space between galaxies. Life, in this sense, is not some magical exception to the rules of the universe. It is a breathtakingly elegant consequence of those rules. The reason this intricate dance works, the reason life could arise from chemistry, the reason complexity could emerge from simplicity, comes down to a deep principle about how energy flows through systems. Living things are not in equilibrium with their environment. If they were, they would be dead. Instead, they are open systems, constantly taking in energy and matter and expelling waste. They create temporary localized islands of order, your body a tree, a bacterial colony, by increasing the disorder or entropy of their surroundings. It's a cosmic bargain. To maintain your own internal order, you must contribute to the overall increase of disorder in the universe. This is why you eat, why you breathe, why you radiate heat. You're a beautifully complex eddy in the vast flowing river of universal entropy. This deep understanding changes our perception of even the most ordinary living things. A blade of grass is no longer just a green stalk. It is a solar-powered chemical factory, converting photons into chemical bonds through processes refined over billions of years. A bird singing in the twilight is not merely making sounds. It is an organism exquisitely tuned to its environment, its every feather and muscle a testament to the patient pressure of survival itself. And you, listening in the quiet of the night, are a living, breathing testament to matter that has learned to contemplate itself, to wonder at the very mechanisms that allow it to exist. There's a particular comfort in understanding that the same principles that govern the smallest atoms also sculpt the most complex organisms. We are not aliens in the universe, but rather an expression of its deepest tendencies. So we've begun to gently trace the intricate, shimmering boundary of what we call life, seeing it not as a list of properties, but as a dynamic, self-sustaining flow of energy and information. But this understanding immediately raises a profound question. If life is ultimately a complex chemical system, how did this delicate dance ever begin? What were the conditions on the early Earth that allowed these first improbable steps to occur? How did the non-living stir itself into the first whisper of life? That question beckons us further into deep time, into a world vastly different from our own, where the very rocks held secrets of creation. The question lingered, a profound echo in the quiet halls of our contemplation. How did this delicate dance ever begin? What conditions on early Earth allowed the non-living to stir into the first whisper of life? We spoke in the previous chapter of the intricate machinery within a cell, the ceaseless molecular ballet that defines life. But before the bustling city, before the language of life could be written and read, there was only a canvas of raw elemental chemistry. Imagine, if you will, stepping back in time, not just millennia, but billions of years, to a world utterly alien, yet undeniably our ancestral home. Picture, outside your mind's window, a planet still nascent, its surface a tumultuous tapestry of volcanic eruptions and searing heat. The sky, not the familiar blue we cherish, but a dense murky shroud of gases, methane, ammonia, water vapour, carbon dioxide, utterly devoid of the free oxygen that now sustains us. Lightning ripped through this primal atmosphere with furious intensity, a relentless crackling energy source. Asteroids and comets, remnants of the solar system's formation, frequently scarred the surface, delivering not only devastating impacts but also, perhaps, crucial icy payloads of water and simple organic molecules from the cosmos itself. This was the early Earth, a crucible of immense power and profound potential. Within this harsh embrace, vast oceans began to coalesce, their waters not merely H2O, but a rich, warm broth, what scientists have poetically termed the primordial soup. Here, amidst the churning tides and volcanic vents, the stage was set for an unprecedented chemical drama. We invite you to gently contemplate this environment, so vastly different from our own, yet fundamentally responsible for every breath we now take. How, in such a place, could the intricate complexity of life possibly emerge? The journey from simple inorganic matter to the first living cell was not a singular, miraculous leap, but rather a patient, incremental unfolding, a series of chemical steps guided by the fundamental laws of physics. We begin with the very building blocks, in that early oxygen-free atmosphere energized by lightning, intense ultraviolet radiation, unfiltered by an ozone layer, and geothermal heat. Simple inorganic molecules began to react: ammonia, methane, water. These humble components under such energetic duress could spontaneously form more complex organic molecules, amino acids, the very constituents of proteins, nucleotides, the letters of genetic code, simple sugars, the fuel and structure, and fatty acids, the potential boundaries of a cell. Consider the famous Miller-Urey experiment, a pivotal moment in our scientific understanding. In a sealed apparatus mimicking early Earth conditions with only water, methane, ammonia, and hydrogen, and a spark to simulate lightning, they observed, to their astonishment, the spontaneous formation of amino acids. This was not life, but it was a profound demonstration. The basic ingredients for life could arise naturally, born from the raw chemistry of the early planet. It was as if the universe, with its vast, indifferent hand, was slowly, patiently assembling the letters of a cosmic alphabet. But letters alone do not make a story. The next crucial step involved these individual letters, these monomers, linking together to form longer chains, or polymers. How did amino acids become proteins, or nucleotides become nucleic acids like RNA and DNA? In the primordial soup, perhaps on the surfaces of mineral clays or around volcanic vents, where conditions allowed for concentration and dehydration, these building blocks could polymerize. Clay minerals, with their intricate layered structures, could have acted as catalysts, binding these molecules and holding them in place, facilitating the chemical reactions that stitched them into longer, more complex chains. Imagine the gentle adherence of these small molecules to a mineral surface, like tiny beads slowly being strung onto a thread. Here we encounter a profound conceptual hurdle, often referred to as the chicken or the egg problem of early life. DNA, the master blueprint of life, stores genetic information, but it requires an army of protein enzymes to replicate itself and express its code. Proteins, the molecular machines that perform nearly all cellular functions, are themselves synthesized according to instructions encoded in DNA via RNA. Which came first? This apparent paradox led scientists to one of the most compelling hypotheses in abiogenesis, the RNA-D world. Contemplate the elegant solution offered by RNA. Unlike DNA, which is a double helix, RNA typically exists as a single strand, making it more versatile. Crucially, certain RNA molecules, called ribozymes, can do something remarkable. They can act as enzymes. This means RNA can both store genetic information and catalyze chemical reactions, including, potentially, its own replication. In the early stages of life, before the evolution of DNA and complex proteins, RNA could have been the star player, a dual-purpose molecule capable of both carrying the genetic message and performing the work of life. It's as if the universe, in its quest for self-organization, discovered a molecule that could write its own instruction manual and then build the tools to follow those instructions. This RNA world offers a plausible pathway for the emergence of self-replicating systems, a crucial precursor to life. Yet even with self-replicating molecules, life requires boundaries. A bustling city needs walls, a distinct inside and outside to maintain its order and protect its intricate processes. This brings us to the formation of protocells. Remember those fatty acids we mentioned formed spontaneously in the early Earth? These molecules have a peculiar and beautiful property. In water, they naturally assemble into spherical structures called vesicles or micelles. These simple lipid membranes forming spontaneously could have encapsulated the nascent RNA molecules and other organic compounds, creating a distinct internal environment separate from the chaotic primordial soup. Imagine these lipid bubbles forming, breaking, reforming, some perhaps accidentally enclosing a particularly efficient self-replicating RNA molecule and its necessary ingredients. Within these fragile spheres, the encapsulated molecules could interact more efficiently, protected from dilution and external interference. This was a critical step towards homeostasis, the ability to maintain an internal stability, a hallmark of life. These protocells, though not yet living in the full sense, represented a monumental leap. The creation of a bounded, self-organizing system capable of evolving. While the primordial soup theory largely focuses on surface reactions, another compelling hypothesis points to the deep ocean. Hydrothermal vents. Picture vast cracks in the Earth's crust, miles beneath the surface, spewing superheated, mineral-rich water. These black smokers create incredibly dynamic environments, rich in chemical energy, and offering protection from the harsh UV radiation and meteor impacts of the surface world. Here, within the intricate, porous structures of mineral deposits, scientists hypothesize that chemical gradients, differences in concentration of ions and molecules, could have driven the synthesis of organic compounds and the formation of protocells. These vents provide a continuous supply of energy and raw materials, a tireless engine for chemical evolution. The transition from non-life to life, from the inert dust of the cosmos to the first stirring of a self-replicating, evolving entity, was not a singular event but a prolonged, patient dance of chemistry over hundreds of millions of years. It was a journey from simple atoms to complex organic molecules, from individual molecules to self-replicating polymers, from unbounded chemistry to encapsulated protocells, and finally to the first true cells capable of metabolism, growth, and evolution. We stand today, you and I, as living testaments to this incredible journey. The very atoms composing our bodies, the elegant dance within our cells, are echoes of that primal chemistry, reassembled stardust that learned to organize itself, to replicate, to evolve. The origin of life remains one of biology's most profound and active areas of research, a testament to the universe's capacity for self-organization and emergent complexity. We may never fully recreate those exact first whispers, but through scientific inquiry, we piece together the story, step by patient step, revealing a narrative more awe-inspiring than any fiction. This journey, from the raw elements to the first flicker of life, reminds us of our deep interconnectedness with the cosmos and with the very fabric of existence. It is a story not of magic, but of the exquisite potential inherent in matter itself, given the right conditions and enough time. And as we contemplate these humble beginnings, we are left with an even greater appreciation for the delicate, intricate tapestry of life that has unfolded since. But what happened next? How did these early, simple life forms diversify and evolve into the astounding biodiversity we see today? This is a question for another quiet contemplation. We left our journey at the cusp of life itself, gazing upon the humble protocell, a fragile bubble of fatty acids cradling the nascent sparks of self-replication. Within its delicate membrane, RNA molecules, those versatile architects, began to whisper the first instructions, to catalyze the first simple reactions. It was a monumental achievement, a breathtaking leap from inert chemistry to the earliest stirrings of biological identity. But how, you might wonder, did this solitary, simple beginning blossom into the riotous, breathtaking diversity that paints our planet today? How did the single, flickering flame of life ignite a forest of forms, from the microscopic wonders hidden in a drop of dew to the towering redwoods that touch the sky, from the silent depths of the ocean to the soaring heights of the eagle? This, dear friends, is where we embark upon perhaps the most profoundly misunderstood, yet utterly foundational concept in all of biology. Evolution by natural selection. It is not a ladder of progress, nor a conscious striving, but a relentless, elegant dance of change, a ceaseless improvisation played out on the grand stage of time. Imagine, for a moment, any group of living things you encounter, perhaps the sparrows fluttering outside your window, the dandelions stubbornly pushing through cracks in the pavement, or even the faces in a bustling crowd. What do you observe? Not perfect uniformity, but a symphony of subtle differences. Each sparrow has a unique feather pattern, each dandelion a slightly different shade of yellow, each human face a distinct arrangement of features. This inherent variation is the first indispensable ingredient in life's grand evolutionary recipe. Where do these whispers of possibility, these infinitesimal deviations, come from? At the heart of it lies the gentle, persistent hum of mutation. Within the very blueprint of life, the DNA or RNA that guides existence, tiny, spontaneous changes occur. A single letter in the vast genetic code might be swapped, deleted, or duplicated. These are often random, unbidden events, like a misplaced comma in a sprawling novel. Many mutations are neutral, some are detrimental, but occasionally a mutation bestows a slight advantage, a new nuance in the organism's interaction with its world. Beyond mutation, the intricate ballet of sexual reproduction shuffles and recombines genetic material from two parents, creating unique combinations in each offspring, amplifying the tapestry of variation with every new generation. Gene flow between populations also adds to this rich genetic mosaic. Now, consider the world these varied beings inhabit. It is not a static, benevolent garden, but an ever-shifting landscape of challenges and opportunities. Resources are finite: food, space, mates. Predators lurk, diseases spread, climates shift. This is the crucible, the environmental sculptor, exerting what we call selection pressure. In this intricate interplay, not all individuals are equally successful in surviving and, crucially, in reproducing. Here lies the elegant heart of natural selection. Individuals with traits that are better suited, or, quote persist, for their specific environment, are more likely to survive long enough to pass those advantageous traits to their offspring. This is not a brutal contest of strength, as the common phrase "quos chesten nein" so often implies. Indeed, this phrase, when taken literally, can be profoundly misleading. Quote Gasquet. In evolutionary terms does not mean the strongest, the fastest, or the most aggressive. It means reproductive success. The ability to produce viable offspring who, in turn, survive and reproduce. A tiny camouflaged insect that blends seamlessly into its surroundings and lays many eggs might be far, quote as Levnalan, than a large flashy one that attracts predators and produces few progeny. The quotes 12 are simply those whose inherited characteristics align most favourably with the demands and opportunities of their current environment, allowing their genetic legacy to continue. Over countless generations, these subtle advantages accumulate. Imagine a population of ancient single-celled organisms in a slightly acidic primordial ocean. If a random mutation gives some individuals a slightly more resilient cell wall, allowing them to thrive better in that acidity, they will, on average, leave more descendants. Their robust cell walls will become more prevalent in the population. The ocean might then grow warmer, and another mutation, perhaps one allowing for better heat tolerance, might arise and spread. This is the gradual incremental accumulation of change that, over millions and billions of years, transforms one form of life into another, giving rise to new species, new adaptations, and ultimately the staggering biodiversity we see today. It is a slow, unhurried river of transformation, carving new paths through the landscape of life. This understanding helps us gently dismantle some of the most persistent misconceptions surrounding evolution. Firstly, evolution is not a linear progression. Up a ladder towards some ideal, quote, 13 with humans at the pinnacle. This anthropocentric view misrepresents the entire process. Instead, envision evolution as a magnificent, sprawling tree of life, constantly branching and rebranching. Every twig, every leaf, represents a lineage that is equally evolved for its particular niche. A bacterium, perfectly adapted to its microscopic world, is no less, quote, 14 genes than a complex mammal. Both have undergone billions of years of adaptation to their respective environments. There is no, quote, fifidnastansis, or still, quote, 16 then, form. There is only differentiation and adaptation. Secondly, evolution is not driven by conscious intent or foresight. Life does not, quote, year 17 to evolve an eye or a wing because it, quote, year 17 1. Instead, random mutations occur, and if one of those mutations, by sheer chance, confers an advantage in a particular environment, it might be favoured by natural selection. The intricate complexity of an eye, for example, did not appear fully formed. It arose through a long series of tiny incremental steps, each one providing a slight visual advantage over its predecessors. From simple light-sensing patches to sophisticated lenses. It's a blind watchmaker, as Richard Dawkins eloquently put it, creating astonishing complexity without a conscious designer. Finally and crucially, individuals do not evolve. You, as an individual, cannot evolve new traits within your lifetime in response to your environment. Your genetic makeup is fixed from conception. It is populations that evolve over successive generations. The frequencies of genes and traits within a population shift as natural selection acts upon the varied individuals within it. The collective genetic heritage of a group changes, not the individual members. The evidence for this grand, unfolding narrative is woven into every thread of existence. We trace it in the Bath fossil record, observing the gradual changes in ancient forms, the emergence of transitional species that bridge vast gaps. We see it in comparative anatomy, marveling at the homologous structures, the five-fingered limb, for example, appearing in the wing of a bat, the flipper of a whale and the arm of a human, all adapted for different functions, yet bearing the unmistakable stamp of a shared ancestry. We uncover it in the profound similarities of molecular biology, where the genetic code itself, the language of life, is almost universally conserved, and the DNA sequences of vastly different organisms reveal their deep familial connections. From the distribution of species across continents, biogeography, to the direct observation of evolutionary change in bacteria and viruses, the story is consistent, compelling, and beautifully intricate. Contemplate for a moment the profound implication of this relentless dance. Every living thing you have ever encountered, from the smallest microbe to the most majestic whale, shares a common ancestor. We are all, in a very real and fundamental sense, family. Trace back the branches of the magnificent tree of life far enough, and you will arrive at a single, ancient root, that very first protocell perhaps, or an even earlier universal common ancestor, a microscopic entity that billions of years ago set in motion the greatest story ever told. Our bodies are reassembled stardust, yes, but our life itself is a continuous unbroken lineage stretching back to that first chemical whisper. This realization is not meant to diminish us, but to expand our sense of belonging, to deepen our reverence for the intricate web of life. We are not isolated entities, but integral threads in a tapestry woven over eons, shaped by the same fundamental forces that sculpted the earliest forms. The concept of evolution, far from being a cold mechanistic theory, is a celebration of life's boundless creativity, its enduring resilience, and its astonishing capacity for transformation. It is the story of how life, once sparked, learned to dance with change, to adapt, to diversify, and to continually reinvent itself in the face of an ever-changing world. As we move forward, we will continue to trace these branches, exploring how life diversified into its incredible kingdoms, how new forms emerged, and how the very mechanisms of life became more intricate and specialized. But for now, let us simply rest in the quiet wonder of this relentless dance, this elegant unfolding that connects every living being, past, present, and future, in a shared ancient heritage. Let's appreciate the quiet power of variation, the subtle pressure of selection, and the boundless patience of time, which together have sculpted the breathtaking beauty of the living world. In our last quiet gathering, we journeyed through the intricate dance of evolution, witnessing how life, born from ancient stardust, diversified through the relentless, beautiful logic of natural selection. We observed how variation within populations, coupled with the selective pressures of an ever-changing world, sculpted organisms into forms exquisitely adapted to their environments. But as we contemplated this vast branching tree of life, a profound question lingered in the air, a whisper carried on the cosmic winds. How, precisely, does one branch become two? How do these myriad forms, these specialized creatures that populate every nook and cranny of our planet, come to be recognized as distinct entities? Today, dear friends, we turn our gaze to one of biology's most fundamental and perhaps most elusive concepts, the species. It is a word we use with casual familiarity, yet its definition, when held up to the light of scientific inquiry, shimmers with unexpected complexity, revealing layers of misunderstanding that often obscure the dynamic beauty of life itself. Imagine, if you will, standing at the edge of a vast ancient forest. You see towering oaks, slender birches, sprawling maples, each distinct, each recognizable. You instinctively categorize them, perhaps calling them different species of trees. This intuitive understanding, this ability to group similar organisms, is deeply ingrained within us. For centuries, naturalists have sought to impose order upon the dizzying array of life, to draw clear lines in nature's vibrant tapestry. But nature, in its boundless creativity, often defies such neat categorization. At its most common and widely accepted, the BSS, biological species concept, BSC, defines a species as a group of populations whose members have the potential to interbreed in nature and produce fertile offspring, but do not produce fertile offspring with members of other such groups. It's a beautiful idea, isn't it? A testament to shared genetic heritage and the promise of future generations. If you and I belong to the same species, it implies a shared lineage, a common thread of genetic information that can be woven anew. We ask you to contemplate this definition for a moment. It suggests a clear boundary, a reproductive wall that separates one species from another. Think of a horse and a donkey. They can mate, yes, producing a mule, but the mule is sterile, unable to carry on the lineage. This, by the BSC, confirms that horses and donkeys are distinct species. The wall holds. Yet, as with so many concepts in biology, the deeper we look, the more nuanced the picture becomes. The BSC, while powerful, is not without its limitations, its quiet ambiguities that challenge our desire for absolute certainty. What of organisms that reproduce asexually, like many bacteria, fungi, or even some plants and reptiles? They do not interbreed, so how do we apply the BSC to them? Or consider the vast fossil record, the silent remnants of life long past. We cannot observe their mating rituals or assess the fertility of their offspring. How do we define species from a collection of ancient bones or impressions in stone? And then there are the fascinating cases of hybridization in nature. Sometimes different species do interbreed, producing fertile offspring, blurring the very lines we try to draw. Certain plant species, for instance, can form fertile hybrids, leading to new species through a process called polyploidy. Even among animals, there are instances where what we once considered distinct species can, under certain circumstances, produce viable, fertile offspring. These exceptions remind us that species is not a fixed, immutable category etched in stone, but rather a dynamic, somewhat fluid concept, a human attempt to categorize a reality that is constantly shifting and evolving. Perhaps the most poetic challenge to our neat categories comes from the phenomenon of ring species. Imagine a chain of populations encircling a geographical barrier like a mountain range. Each population can interbreed with its immediate neighbors. Yet when you trace the chain all the way around, the populations at the ends of the ring, though connected by a series of interbreeding neighbors, cannot interbreed with each other. They have diverged sufficiently over the long journey. Where, then, do we draw the line? At what point does a gradual, imperceptible shift become a distinct species? The ring species illustrates that evolution does not always proceed in discrete jumps. Sometimes it is a slow, continuous gradient, a spectrum of change rather than a sudden break. This leads us to the heart of the matter: speciation. The very process by which one species splits into two or more distinct species. It is the engine of biodiversity, the mechanism that has populated our world with its breathtaking array of life. At its core, speciation requires reproductive isolation. The development of barriers that prevent gene flow between populations. Once gene flow ceases, isolated populations are free to diverge genetically, accumulating different mutations, experiencing different selective pressures, and undergoing different patterns of genetic drift. Over vast stretches of time, these differences can become so pronounced that, even if the populations were to meet again, they could no longer interbreed successfully. Let us explore two primary pathways to speciation. The first, and arguably most common, is allopatric speciation. Here, allopatric means other country, hinting at the crucial role of geographic isolation. Imagine a single, thriving population of, say, squirrels. Then, a geological event occurs, a new river carves through their habitat, or a mountain range slowly rises, or a glacier advances, physically separating the population into two isolated groups. Now these two groups, living in different countries, face different environmental challenges. One side of the river might have harsher winters, favouring thicker fur. The other side might have different predators, leading to different camouflage patterns. Over countless generations, genetic changes accumulate independently in each group. Mutations arise, natural selection acts differently, and genetic drift randomly shifts allele frequencies. Eventually, even if the geographic barrier were to disappear and the two squirrel populations were to meet again, they would no longer recognize each other as potential mates, or their offspring would be infertile. They have become distinct species. Think of the snapping shrimp on either side of the Isthmus of Panama. Before the isthmus rose, a single population of shrimp likely spanned the open ocean. As the land bridge formed, separating the Pacific and Atlantic Oceans, the shrimp populations were divided. Over millions of years they diverged. Today, if you place a Pacific snapping shrimp with an Atlantic snapping shrimp, they will snap aggressively at each other, but they will not mate. Their reproductive isolation is complete, a testament to the power of geographic separation. The second pathway, more subtly profound, is sympatric speciation, where sympatric means same country. Here, new species arise within the same geographic area, without any obvious physical barrier. How can this be? It often involves mechanisms that create reproductive isolation within a continuous population. One powerful example, especially prevalent in plants, is polyploidy, where an organism ends up with more than two sets of chromosomes. This can happen through errors in cell division. A polyploid individual is often instantly reproductively isolated from its diploid, two sets of chromosomes, ancestors, as their offspring would typically be sterile. Thus, a new species can arise in a single generation. Other forms of sympatric speciation can involve habitat differentiation, where subsets of a population begin to exploit different resources or niches within the same area. Consider a population of insects that feed on a particular type of fruit. A mutation might arise that allows some individuals to feed on a different type of fruit that grows nearby. If these fruit-specific preferences lead to mating primarily among individuals that prefer the same fruit, then over time, two reproductively isolated populations could emerge, even though they live side by side. Sexual selection can also play a role, particularly in animals, where individuals choose mates based on specific traits, leading to divergence in mating preferences that eventually isolate populations. The development of these reproductive barriers is key to both allopatric and sympatric speciation. These barriers can be prezygotic, preventing the formation of a zygote, a fertilized egg. This might involve habitat isolation, different species live in different places, temporal isolation, they breed at different times, behavioral isolation, different courtship rituals, mechanical isolation, mating parts don't fit, or gametic isolation, sperm cannot fertilize egg. Or the barriers can be post-zygotic, acting after a zygote has formed, such as reduced hybrid viability, hybrid offspring don't survive, reduced hybrid fertility, mules, or hybrid breakdown. First-generation hybrids are fertile, but subsequent generations are not. Each of these mechanisms acts as a lock, preventing the flow of genetic information and allowing populations to drift apart, eventually becoming distinct species. As we contemplate these intricate processes, we begin to grasp the sheer, breathtaking scale of biodiversity on Earth. Every act of speciation adds a new thread to the planet's living tapestry. We have identified nearly 2 million species, yet estimates suggest there could be anywhere from 5 million to 100 million species in total, with vast numbers of bacteria, fungi, and insects still awaiting discovery. This incredible variety is not merely a beautiful spectacle. It is the very foundation of a healthy ecosystems, providing stability, resilience, and a vast library of genetic information that holds untold secrets and potential solutions for our own challenges. Each species, no matter how small or seemingly insignificant, plays a role in the intricate web of life, contributing to the delicate balance that sustains us all. Yet, despite this profound understanding, misconceptions about species and biodiversity persist, clouding our perception. One common misunderstanding is the concept of, quote, 20. You may hear people speak of a, quote, between apes and humans, for instance. This idea stems from a linear view of evolution, picturing life as a ladder with, or a, quote, 30 forms at the bottom, and cast, quote, 13 forms, like humans, at the top. But as we have learned, evolution is not a ladder. It is a sprawling, branching tree. Every organism alive today, and every fossilized ancestor is a Consi, quote, 32 in its own lineage. There is no single, transitional, quote, 30 through St that perfectly bridges two groups, because evolution is a continuous process of branching and divergence, not a direct progression. We are not the pinnacle of evolution. We are simply one twig on an enormous ancient tree, exquisitely adapted to our own particular niche, just as every other species is to theirs. Another misconception is that species are fixed, unchanging categories, perfectly stable and immutable since their creation. This view contradicts everything we know about evolution. Species are not static. They are dynamic, ever-evolving populations, constantly adapting, splitting, and, sadly, sometimes going extinct. The boundaries between species, as we've discussed with ring species and hybridization, can be fuzzy, reflecting the continuous nature of evolutionary change. And finally, the notion of human superiority often leads us to devalue other forms of life. But in the grand sweep of biological time, no species is more evolved than another. A bacterium that has thrived for billions of years is just as evolved in its own context as a complex mammal. Each is a masterpiece of adaptation, a testament to the power of natural selection to shape life to its environment. We are interconnected, woven into the same fabric of existence, all descendants of that ancient stardust, all part of this magnificent ongoing story. As the quiet hum of our contemplation settles, we invite you to look out into the world with new eyes. See the diversity not as a collection of separate static entities, but as a living, breathing testament to the ceaseless creativity of evolution. Every bird's song, every rustle of leaves, every vibrant bloom is a chapter in the epic tale of speciation, a whisper from countless generations of adaptation and divergence. To understand species is to understand the very engine of life's boundless variety, to appreciate the delicate dance between isolation and interaction that has given rise to the world we inhabit. And with this understanding perhaps comes a deeper sense of wonder, a more profound appreciation for the interconnectedness of all life, and a renewed commitment to safeguarding this precious, evolving tapestry. In our next gathering, we shall delve into the intricate relationships that bind these diverse species together, exploring the fascinating world of ecosystems and the delicate balance that sustains them. We have journeyed through the vastness of cosmic time, tracing the very atoms that form us back to their stellar nurseries, and then witnessed the first whispers of life stirring in Earth's primordial crucible. We have marvelled at the slow, patient dance of evolution shaping nascent forms into the incredible diversity we see today, and finally, pondered the intricate mechanisms that draw lines between these myriad life forms, giving rise to distinct species. Now let us expand our gaze once more, beyond the individual species, beyond the distinct lineage, to see how these countless threads of life are woven together into an even grander tapestry, the living, breathing, interconnected world of an ecosystem. Imagine, if you will, stepping into a forest, or perhaps a coral reef, or even simply observing the life thriving in a patch of soil beneath your feet. What do you notice? It is not merely a collection of isolated entities, but a vibrant, bustling community where every organism, from the tallest tree to the most microscopic bacterium, is engaged in a ceaseless dialogue with its surroundings and with one another. This web of interactions, this dynamic interplay between living beings and their non-living environment, the sunlight, the water, the minerals, the air, is what we call an ecosystem. At the heart of understanding these intricate relationships lies a concept often misunderstood, yet profoundly illuminating: the ecological niche. When we speak of a niche, we are not merely referring to an organism's physical address, its habitat. No, the niche is far more profound. It is, in essence, an organism's profession within its community, its unique role, its entire way of life. It encompasses everything: what it eats, what eats it, where it lives, when it's active, how it reproduces, what resources it uses, and how it interacts with every other living and non-living component of its environment. It is the sum total of all its biotic and abiotic requirements and tolerances. Consider a majestic oak tree. Its niche isn't just the forest floor. Its niche includes its need for sunlight, water, and specific soil nutrients, its role as a producer, converting solar energy into biomass, its relationship with the fungi on its roots, the insects that feed on its leaves, the birds that nest in its branches, the squirrels that bury its acorns, the decomposers that break down its fallen leaves. Every single one of these interactions defines a part of its ecological niche. We can even distinguish between a fundamental niche, the entire set of conditions under which a species could survive and reproduce if there were no competition or predators, and its realized niche, the actual set of conditions and resources it uses in the presence of those competitors and predators. Often the realized niche is a smaller, more constrained version of the fundamental one, a testament to the pressures of existing within a complex community. This concept of the niche immediately brings us to one of the most fundamental forces shaping ecosystems: competition. When two or more organisms require the same limited resources, be it food, water, light, space, or mates, they inevitably compete. This competition can be intraspecific, occurring among individuals of the same species, perhaps for territory or reproductive partners, or it can be interspecific, taking place between individuals of different species vying for shared resources. Think of two different species of warblers foraging for insects in the same tree, or various plant species competing for sunlight on the forest floor, or predators struggling to catch the same prey. It is here that we encounter a powerful ecological principle, the competitive exclusion principle. This principle suggests that if two species have exactly the same ecological niche and are competing for the exact same limited resources, one will eventually outcompete the other, leading to the local extinction of the less successful species. Nature, in its elegant efficiency, abhors a vacuum, but it also dislikes redundancy in its most resource-intensive roles. However, this does not mean that every ecosystem is a brutal, constant battle to the death. Far from it. Often, species that appear to share similar resources manage to coexist through a process called resource partitioning. They might specialize in slightly different food sources, forage at different times of day, or utilize different parts of a habitat. Imagine several species of birds all feeding on insects in the same tree. One might forage on the upper branches, another on the lower trunk, and a third on the inner leaves. They have partitioned the resource, effectively occupying slightly different realized niches, thus reducing direct competition and allowing for peaceful coexistence. This partitioning is a beautiful demonstration of evolution in action, driven by the subtle pressures of competition to find unique ways of life within the larger community. Yet to focus solely on competition would be to paint an incomplete, indeed, a misleading picture of life's intricate dance. While competition is undeniably a powerful force, it is balanced and often overshadowed by an equally vital, if not more pervasive force: cooperation, or more broadly, symbiotic relationships. It is a common misconception that nature is solely, quote, 37, a brutal arena where only the strongest survive in isolation. This perspective overlooks the profound beauty and necessity of interdependence. Symbiosis, meaning just, quote, 38 describes close and long-term interactions between different biological species. These relationships can take many forms. We observe mutualism, when both species benefit from the interaction. Consider the vibrant coral reefs, veritable underwater cities teeming with life. Their very foundation relies on a mutualistic partnership between coral polyps and microscopic algae called zooxanthellae. The algae live within the coral tissues, providing the coral with photosynthetic products, sugars, while the coral offers the algae a protected environment and compounds they need for photosynthesis. Without this partnership, neither could thrive as they do. Or think of the tiny, unseen world within our own bodies, the vast communities of bacteria in our gut that help us digest food and synthesize vitamins, receiving a warm, nutrient-rich home in return. This is mutualism on a grand, intimate scale. Then there is commensalism, where one species benefits and the other is neither significantly helped nor harmed. Barnacles, for instance, often attach themselves to whales, gaining a mobile home and access to nutrient-rich waters as the whale swims, while the whale appears largely unaffected. Or consider the cattle egrets that follow grazing livestock, feeding on the insects stirred up by the animal's movements. The egrets benefit from an easy meal, and the cattle are generally indifferent. Finally, we have parasitism, an interaction where one species, the parasite, benefits at the expense of the other, the host. Ticks feeding on the blood of a deer, tapeworms living in an animal's intestine, or mistletoe drawing nutrients from a host tree. These are all examples of parasitism. While it might seem negative from a human perspective, parasites are an integral part of ecosystem dynamics, influencing host populations and even driving co-evolutionary arms races that lead to incredible adaptations in both parasite and host. They are not simply bad guys, but threads in the complex web, often regulating populations and maintaining balance. These interactions, competition and the various forms of symbiosis, are the invisible threads that weave together the intricate fabric of life, defining who eats whom, who helps whom, and who lives where. They dictate the flow of energy and the cycling of nutrients through an ecosystem. This brings us to the concept of trophic levels and higher food webs. At the base of every food web are the maya producers, primarily photosynthetic organisms like plants and algae, which convert sunlight into chemical energy. They are the foundation of life, creating the energy currency for almost all other organisms. Above them are the consumers, herbivores, primary consumers, that eat producers, carnivores, secondary and tertiary consumers, that eat other consumers, and omnivores that eat both. And finally, the essential decomposers, bacteria, fungi and detritivores, which break down dead organic matter, recycling vital nutrients back into the soil or water, making them available for the producers once more. A food chain is a simplistic linear representation of who eats whom. But the reality is far more complex and interconnected, forming a vast branching food web, where organisms often feed on multiple species and are themselves prey for several others. Every organism in an ecosystem is a node in this immense dynamic web of energy transfer and nutrient cycling. Remove one strand, and the entire web shivers, sometimes subtly, sometimes dramatically. When we truly grasp the concept of ecological niches and the spectrum of interactions from fierce competition to profound mutualism, we begin to see the world with new eyes. We realize that the notion of humans separate from nature is a profound illusion. We are not external observers, but integral, deeply embedded participants in these very same ecological webs. Our actions, our consumption, our waste, our presence, all reverberate through these interconnected systems, affecting countless other niches and relationships. The bustling city of life that is an ecosystem is not a static painting, but a continuously evolving, breathing entity. Its complexity is mind-boggling, its resilience awe-inspiring, and its delicate balance a constant source of wonder. To understand a species is to understand its niche, its relationships, its place in the grand narrative of energy and life. We are invited to move beyond simple categories and embrace the fluid, interdependent nature of existence. As we conclude this chapter, let us carry with us a profound appreciation for the invisible threads that bind all life. Look outside your window, or simply observe the dust motes dancing in a sunbeam, and contemplate the countless, unseen interactions happening all around you, all the time. Every breath you take, every bite you eat, every living thing you encounter is a testament to this magnificent interconnected dance of life. It is a symphony of niches, a ballet of relationships, a testament to the fact that in biology, nothing truly exists in isolation. Everything is connected, profoundly and beautifully so. And in this understanding, we find not only scientific truth, but also a deeper sense of belonging and responsibility within this breathtaking living world. As we journeyed through the intricate tapestry of ecosystems in our last contemplation, we marveled at the delicate dance of niches, the competitive ballet, and the profound embrace of symbiotic relationships. We traced the vibrant threads of energy as they wove through trophic levels, from the patient producers drawing sustenance from the sun to the tireless decomposers, returning life's building blocks to the earth. We understood deeply that every living thing, from the smallest bacterium to the towering redwood, is inextricably linked within these magnificent self-sustaining webs. Yet, there is a common whisper in our collective consciousness, a comforting belief that nature, left to its own devices, exists in a pristine, unchanging equilibrium. We often imagine a static, perfect balance, a tableau vivant of perpetual serenity. But if we allow ourselves to look closer, to listen to the ancient rhythms of our planet, we begin to perceive a truth far more dynamic, more profound, and perhaps even more awe-inspiring. Ecosystems are not static portraits, but living, breathing symphonies, constantly composing and recomposing themselves through cycles of change, disturbance, and renewal. Let us together gently challenge this notion of unchanging balance. Imagine, if you will, a landscape scarred by a sudden intense event, perhaps a volcanic eruption, blanketing vast stretches of land in sterile ash and cooled lava. Or consider a majestic forest, brought low by the searing breath of a wildfire, leaving behind only charred trunks and a blackened earth. In these moments of profound alteration, where life seems utterly vanquished, what then? Does the ecosystem simply cease to exist? Or does it, with an astonishing resilience, begin a new story? This brings us to one of the most fundamental, yet often overlooked, principles of ecological life: ecological succession. It is the gradual, sequential process by which communities of organisms colonize an area and are then replaced by other communities over time, until a relatively stable, mature community is established. It is the Earth's patient, persistent act of rebuilding, of reweaving its living fabric. We distinguish between two primary forms of this grand narrative. Primary succession unfolds in areas entirely devoid of life, where no soil exists, a fresh canvas of bare rock, newly formed volcanic islands, or land exposed by retreating glaciers. Imagine the sheer audacity of the first pioneers, lichens and mosses, tenacious and humble, clinging to barren stone. They are the true adventurers, secreting acids that slowly, painstakingly break down the rock, mingling with their own decaying matter to form the very first nascent layers of soil. Over centuries, these humble beginnings paved the way for grasses, then shrubs, and eventually the magnificent trees of a mature forest. It is a slow, quiet miracle, a testament to life's unwavering resolve to find purchase, to create possibility where none existed. Then there is secondary succession, a process that occurs in areas where a pre-existing community has been disturbed or removed, but the soil remains intact. Think of the forest after a fire or an abandoned agricultural field. Here, the rebuilding begins with a significant advantage: the rich legacy of the soil, brimming with dormant seeds and microbial life. Weeds and grasses quickly colonize, stabilizing the soil and providing shade. Soon, fast-growing shrubs emerge, followed by a stiff 42 wound, tree species that thrive in abundant sunlight. Gradually, these give way to more shade-tolerant, slower-growing species, until, over decades or centuries, a forest much like the original might stand once more. This recovery, while still a long unfolding, is often swifter, a testament to the enduring memory held within the earth itself. The endpoint of this succession was once termed a Gizskan, quote, 43, a stable, self-perpetuating ecosystem, thought to be in perfect balance with its environment. But here too our understanding has deepened. We now recognize that even these mature communities are not truly static. They are dynamic, ever-shifting mosaics, constantly adapting to minor disturbances, subtle climatic shifts, and the ongoing dance of life and death within their boundaries. The quote 44 is less a fixed destination and more a state of dynamic equilibrium, a perpetual gentle sway rather than an absolute stillness. This brings us to a crucial, often misunderstood aspect of ecosystem dynamics, the role of disturbance. We tend to view disturbances, wildfires, floods, pest outbreaks, volcanic eruptions, even hurricanes, as purely destructive forces, interruptions to nature's tranquility. And indeed, they can be devastating in their immediate impact. Yet, from a broader ecological perspective, natural disturbances are not merely destructive, they are also profoundly creative. Consider the wildfire. While it consumes, it also clears, opening the forest canopy to sunlight, returning nutrients to the soil, and stimulating the germination of seeds that require heat to sprout. Many species, in fact, have evolved to depend on these periodic disturbances. The lodgepole pine, for example, holds its seeds in resin-sealed cones that only open in the intense heat of a fire. Without fire, its regeneration cycle would be broken. Floods enrich floodplains with nutrient-laden sediments, revitalizing agricultural lands and wetlands. Windstorms create gaps in forests, allowing new light to reach the forest floor, fostering biodiversity by creating diverse microhabitats. These natural disturbances are the sculptors of landscapes, the drivers of succession, ensuring that ecosystems remain vibrant, diverse, and adaptable. They prevent stagnation, recycle resources, and create opportunities for new life to flourish. They are not anomalies, they are intrinsic to the planet's living processes, a rhythmic pulse of destruction and rebirth. How, then, do ecosystems cope with these inevitable shifts and shocks? We speak of their resistance and resilience. Resistance is the ability of an ecosystem to resist disturbance and remain unchanged. A highly resistant ecosystem might shrug off a mild drought or a minor pest outbreak without significant alteration. Resilience, on the other hand, is the capacity of an ecosystem to recover after a disturbance, to bounce back to its original state, or to an alternative, healthy state. A resilient forest, though ravaged by fire, might regenerate relatively quickly, its intricate web of life reassembling itself over time. What factors contribute to an ecosystem's resistance and resilience? Often it is Jiriam biodiversity, the sheer variety of life within it. A diverse ecosystem is like a complex machine with many redundant parts. If one species is lost or one pathway is disrupted, others can step in to fill the void. A diverse food web with multiple connections is less likely to collapse if one link breaks. The intricate web of interactions, the multitude of species performing similar functions, acts as a buffer against change, lending strength and flexibility to the entire system. But now, let us turn our gaze to a different kind of disturbance, one that is not woven into the ancient fabric of Earth's natural cycles, but rather one that has emerged with unprecedented speed and scale. Human impact. We, as a species, are not separate from these ecological dynamics. We are an integral and increasingly dominant part of them. The misconception that we stand outside of nature, observing it from a detached distance, is perhaps one of the most profound misunderstandings of our time. Every breath we take, every resource we consume, every waste product we generate ripples through the global ecosystem. Our impact often differs from natural disturbances in several critical ways. Natural events are generally localized and cyclical, allowing for recovery and adaptation. Human-induced changes, however, are frequently global in scope, rapid in pace, and often introduce novel pressures to which ecosystems have no evolved resistance or resilience. Consider habitat destruction and fragmentation, where vast swathes of forest or grassland are converted for agriculture, urban development, or infrastructure. This is not merely a localized fire, it is a permanent erasure, often leaving behind isolated pockets of habitat too small to sustain viable populations, severing the very connections that allow species to thrive and adapt. Think of pollution, chemicals, plastics, and greenhouse gases released into the air, water, and soil. These are not the natural nutrient cycles of a flood. They are foreign substances, often toxic, disrupting delicate biochemical processes and accumulating in food webs. The warming of our planet, driven by anthropogenic greenhouse gas emissions, is altering climates at a rate far exceeding the capacity of many species and ecosystems to adapt, pushing them beyond their thresholds of resilience. These profound alterations pose an immense challenge. They test the very limits of ecological resilience, sometimes pushing ecosystem past a tipping point from which they cannot recover to their former state, instead transitioning into degraded or entirely novel configurations. The quiet beauty of a coral reef, vibrant with life, can bleach and die under sustained thermal stress, transforming into a barren calcium carbonate graveyard. A rich, biodiverse forest might be replaced by a monoculture plantation, capable of little resilience against disease or further disturbance. Yet understanding these dynamics also empowers us. Recognizing our place within the ecosystem, not separate from it, shifts our perspective from detached observer to responsible participant. It illuminates the profound interconnectedness that means our well-being is inextricably linked to the health of the natural world. It underscores the urgency of our conservation and our restoration efforts, not as attempts to freeze nature in a static moment, but as informed interventions to mitigate our destructive impacts, to assist ecosystems in their natural processes of renewal, and to protect the biodiversity that is the very engine of resilience. So, as you look out upon the world today, perhaps at the quiet steadfastness of a tree or the bustling activity of a city park, remember the hidden narrative of change and continuity. Remember that the balance of nature is not a fragile stasis, but a vigorous dynamic equilibrium, constantly shaped by the ebb and flow of life, death, and renewal. And remember that we too are part of this grand unfolding story, with the capacity to understand, to cherish, and to contribute to its ongoing, magnificent composition. Let this understanding foster not despair, but a deeper sense of wonder, a quiet call to humility, and a renewed commitment to our shared biological heritage. We have journeyed through the vast, interconnected tapestry of life, from the reassembled stardust of our origins to the dynamic dance of ecosystems. We have seen how life, in its myriad forms, continually builds, breaks down and rebuilds, responding to the subtle and dramatic shifts of its environment. We spoke of disturbances, of resilience, and of the profound, often challenging impact of our own species upon this delicate balance. But as we consider the grand scale of ecological change, a more intimate question arises. How do individual organisms, including ourselves, navigate the ceaseless currents of an ever-changing world? How do we maintain our very being amidst such flux? Let us draw our attention inward, to the miraculous mechanisms that allow each living entity to persist, to thrive, and to respond. We speak now of the subtle art of self-regulation, of the intimate dance between an organism and its immediate surroundings, and of the deeper generational shaping that allows life to endure across eons. Recall, if you will, our initial contemplation of life's fundamental characteristics in Chapter 1. Among them, we noted homeostasis, the ability to maintain a stable internal environment despite fluctuations in the external world. At first glance, this might seem a simple concept, a static equilibrium. But let us delve deeper, for homeostasis is anything but static. It is a dynamic, vigilant process, a continuous negotiation with reality. Imagine, for a moment, the intricate workings within your own body. Outside, the temperature might rise or fall, the air might thicken with humidity or dry with a biting wind. Yet within you, a constant, precise temperature of around 98.6 degrees 37 dn is meticulously maintained. Your blood pH remains within a narrow, life-sustaining range. The concentration of glucose, salts, and oxygen in your bloodstream is rigorously regulated. This is not passive endurance. It is an active, ongoing effort, a symphony of biological processes orchestrated to preserve the delicate conditions necessary for life's chemistry to unfold. How is this marvel achieved? Primarily through elegant, negative feedback loops. Think of the thermostat in your home. When the temperature deviates from your set point, the thermostat detects the change and triggers a response. The furnace kicks on if it's too cold, the air conditioning if it's too hot, until the desired temperature is restored. Biological systems operate on a similar principle, but with infinitely greater complexity and sensitivity. Consider your body temperature. If you step out into a chilly autumn morning, your internal sensors detect a slight drop in temperature. This signal races to your brain, which then initiates a cascade of responses. Blood vessels near your skin constrict to reduce heat loss, muscles begin to shiver to generate heat, and metabolic rate might subtly increase. As your body temperature rises back to its set point, these corrective actions gradually cease. Conversely, if you exert yourself on a warm day, your body detects rising temperature and responds by dilating blood vessels to release heat and initiating sweating, which cools the skin through evaporation. The crucial aspect here is negative. The response negates or reverses the initial stimulus, bringing the system back to its optimal range. While negative feedback loops are the guardians of stability, a less common but equally vital mechanism exists: positive feedback loops. Unlike their negative counterparts, these loops amplify the initial stimulus, driving a process to completion. Think of childbirth. The contractions push the baby against the cervix, which in turn stimulates the release of oxytocin, intensifying the contractions further until the baby is born. Or consider blood clotting. Platelets adhere to a wound, releasing chemicals that attract more platelets, forming a plug that seals the injury. These are transient, self-limiting processes, crucial for specific, often rapid, biological events. So, homeostasis is not a static state, but a dynamic equilibrium, a constant active vigilance. It is the very essence of an organism's ability to maintain its integrity in the face of change. But what of the longer-term changes, the shifts in environment that persist for more than a day or a season? How does life adapt beyond the immediate physiological adjustments? Here, we encounter a distinction often blurred in common understanding: the difference between adaptation and acclimatization. Both terms speak to an organism's ability to cope with its environment, but they operate on vastly different timescales and mechanisms. Let us first revisit adaptation. We touched upon this in Chapter 3, when we explored the grand narrative of evolution by natural selection. An adaptation, in the biological sense, is a heritable trait that has evolved over generations within a population because it enhances an organism's survival and reproductive success in a specific environment. These are the specialized beaks of finches, perfectly suited for cracking particular seeds, the camouflage of a chameleon, allowing it to blend seamlessly with its surroundings, the thick fur of a polar bear providing insulation in frigid climes, or the deep roots of a desert plant reaching for precious water. Crucially, adaptations arise from random genetic mutations that, by chance, confer a benefit in a particular environmental context. Individuals possessing these advantageous traits are more likely to survive, reproduce, and pass on their genes. Over many generations, these advantageous genes become more prevalent in the population, gradually shaping the species. An individual organism cannot choose to adapt, nor can it acquire a new adaptation within its lifetime and pass it on. The process is slow, unconscious, and operates on the level of the population's gene pool. It is the very engine of evolutionary change, sculpting life to fit the contours of its world. Now, let us introduce acclimatization. This is a physiological adjustment an individual organism makes within its own lifetime in response to environmental changes. Unlike adaptation, acclimatization is not heritable. It does not involve a change in the organism's genetic makeup that can be passed to offspring. Instead, it is a reversible, temporary modification of an individual's phenotype, its observable characteristics, to better cope with a new set of conditions. Imagine a person from sea level traveling to a high-altitude mountain range. Initially, they might experience shortness of breath and fatigue, but over several days or weeks, their body acclimatizes. They produce more red blood cells to carry oxygen more efficiently, their breathing rate adjusts, and their heart rate might change. These are physiological responses, not genetic ones. If that person returns to sea level, their body will gradually revert to its previous state. Similarly, a plant grown in shade might develop larger, thinner leaves to capture diffuse light, while the same species grown in full sun might produce smaller, thicker leaves to reduce water loss. This is acclimatization to light intensity. Or think of an animal growing a thicker coat of fur in response to the onset of winter, only to shed it in spring. These are individual responses, allowing for immediate flexibility. The distinction is vital. Adaptation is a genetic, generational process that changes the species. Acclimatization is a physiological, individual process that changes the organism for a time. One reshapes the blueprint, the other adjusts the building's ventilation. However, the line between these two concepts becomes wonderfully nuanced when we introduce phenotypic plasticity. This is the capacity of a single genotype, an organism's genetic makeup, to produce different phenotypes, observable traits, in response to varying environmental conditions. In essence, phenotypic plasticity is the ability to acclimatize, and this ability itself is an adaptation. Consider a species of water flea, Daphnia. In the presence of predators, Daphnia can develop protective helmets and spines. When predators are absent, they do not. The Daphnia doesn't, quote 47, to grow a helmet. Rather, its genes contain the instructions to produce a helmet. If certain environmental cues, like predator chemical signals, are present, this flexibility, this capacity to alter its form based on environmental signals, is a highly adaptive trait. It allows the organism to conserve energy when the defense isn't needed, but quickly deploy it when danger arises. Another striking example is seen in many plants. A single plant with the same genetic code might produce different leaf shapes, sizes, or thicknesses depending on whether its branches are in full sun or deep shade. The genes don't dictate one specific leaf, but rather the rules for leaf development under different conditions. This phenotypic plasticity allows the plant to optimize photosynthesis and water retention across its own body or in varying microclimates. So, while acclimatization is an individual's immediate response, phenotypic plasticity is the evolved genetic capacity for that individual response. It's like having a versatile toolbox encoded in your DNA, allowing you to select and deploy different physiological or morphological tools depending on the specific environmental challenge you face. This capacity for flexibility is itself a product of natural selection, favored in environments that are variable or unpredictable. As we bring these concepts together, we begin to see life not as a fragile entity, clinging precariously to existence, but as a marvel of dynamic responsiveness. Homeostasis ensures internal stability moment by moment. Acclimatization provides short-term, reversible flexibility for the individual. Phenotypic plasticity is the evolved genetic blueprint that confers the ability to acclimatize. And adaptation, the grand sculptor, shapes populations over countless generations, embedding advantageous traits into the very fabric of species. We are not passive recipients of environmental change. We are, each of us, and every living thing around us, a testament to active, dynamic responsiveness. Life is a constant negotiation, a breathtaking display of intricate feedback loops, subtle physiological shifts, and profound genetic legacies. It is a continuous conversation between organism and environment, spoken in the language of chemistry, structure, and behavior. Contemplate this for a moment. The resilience we observe in the grand ecosystems, the ability of a forest to regenerate after fire, is mirrored in the quiet, persistent work within every cell, every tissue, every organism. From the microscopic dance of molecules to the vast sweep of evolutionary time, life is defined by its ability to respond, to adjust, to persist, and to transform. It is a testament to the elegant self-organizing power of the universe, a power that flows through us all. But how do these intricate instructions for response and transformation, these blueprints for adaptability, actually work at their most fundamental level? How is this information stored, expressed, and passed down? This leads us to the heart of heredity, the very language of life itself. We have journeyed through the vast unfolding narrative of life, observing its intricate dance from the primordial stirrings to the complex dynamic ecosystems that breathe and shift around us. We have peered into the resilience of species and the remarkable ability of individuals to acclimatize, to make subtle adjustments within their own lifetimes, guided by an inherent flexibility. Yet, a fundamental question lingers, a whisper in the quiet chambers of our understanding. How are these blueprints for life, these capacities for adaptation and transformation, encoded? How are the instructions for building a beating heart, for sensing the subtle shifts in the wind, and for unfolding a leaf towards the sun, stored and meticulously passed from one generation to the next? Let us now turn our gaze inward, into the very core of what makes each living thing uniquely itself, and yet profoundly connected to all others. We invite you to contemplate the elegant language of life, the script written in molecules that orchestrates the symphony of existence. We speak, of course, of heredity and the astonishing molecule at its heart, DNA. Imagine, if you will, not just a blueprint, but an entire library, meticulously catalogued and cross-referenced, containing every instruction for every cellular process, every protein, every potential form and function an organism might possess. This is, in essence, what the DNA within each of your cells represents. Deoxyribonucleic acid, a name that rolls off the tongue like a complex incantation, is the master instruction manual, the enduring code that binds all life. We often picture DNA as the iconic double helix, a spiraling ladder of incredible elegance. Each rung of this ladder is formed by a pair of chemical bases, adenine, A, always pairing with thymine, T, and guanine, G, always pairing with cytosine, C. It is the specific sequence of these base pairs along the ladder that constitutes the genetic code, a universal language understood by nearly every living cell on Earth, from the smallest bacterium to the tallest redwood, from the deepest sea vent worm to the human contemplating these words. This language, with its four-letter alphabet, dictates the construction and operation of life. Within this vast molecular library, specific sections of this DNA sequence are designated as genes. We might think of a gene as a single, complete recipe within the larger cookbook, providing the instructions for building a specific protein or for regulating another gene's activity. These proteins, in turn, are the workhorses of the cell carrying out myriad functions, forming structural components, catalyzing biochemical reactions, transporting molecules, and much more. Thus, genes are the fundamental units of heredity, dictating the traits an organism will express. But life, as we know, is never so simple as a single recipe. For many genes, there exist different versions, like variations on a theme for a particular recipe. These variations are called persist alleles. Consider for a moment the gene for eye color. While the gene itself dictates the presence of eye pigment, different alleles of that gene might specify shades of brown, blue, or green. You, like all sexually reproducing organisms, inherit 2 copies of most genes, one from each parent. These 2 copies may be identical alleles, or they may be different. This interplay of alleles is where the beautiful tapestry of individual variation begins to unfold. This brings us to a crucial distinction, one often blurred in our everyday understanding, the difference between genotype and phenotype. Your genotype refers to the specific combination of alleles you possess for a particular gene, or, more broadly, your entire genetic makeup. It is the invisible code, the precise sequence of letters in your internal instruction manual. Your phenotype, on the other hand, is the observable expression of that genotype. It is the visible trait, your eye color, your height, the texture of your hair, or even less visible characteristics like your blood type or your susceptibility to certain diseases. It is vital to understand that your phenotype is not solely determined by your genotype. Remember our discussion of phenotypic plasticity at the previous chapter. The environment plays a profound role in shaping how your genes are expressed. A plant might have a genetic predisposition, genotype, for tall growth, but if it is deprived of sunlight, water, or nutrients, its actual height, phenotype, will be stunted. Similarly, while genetics contribute significantly to human height, nutrition during childhood, for instance, can greatly influence whether that genetic potential is fully realized. We carry the potential within our genes, but the world around us helps to sculpt its final form. In our early explorations of heredity, we often encounter the foundational work of Gregor Mendel, who, through his meticulous pea plant experiments, first elucidated the concepts of dominant and recessive alleles. If you inherit one dominant allele and one recessive allele for a particular gene, the dominant allele's trait will typically be expressed in your phenotype, masking the presence of the recessive allele. The recessive trait will only appear if you inherit 2 copies of the recessive allele. This concept, while revolutionary, is frequently misunderstood. We might mistakenly assume that, quote, fortius means, quote, 49 or, quote, 50. This is far from true. Consider polydactyly, the condition of having extra fingers or toes. This trait is caused by a dominant allele, yet it is relatively rare in the human population. Conversely, having 5 fingers, the Yasati, quote, 51oi condition, is associated with a recessive allele, yet it is overwhelmingly common. The terms fias, quote, 52 and, quote, faithrestas simply describe how alleles interact in expressing a trait, not their prevalence or inherent value. Moreover, the complexity of life rarely conforms to such simple, clean-cut categories. While Mendelian genetics provides a fundamental framework, many traits exhibit more nuanced patterns of inheritance. Sometimes neither allele is fully dominant, leading to incomplete dominance, where the phenotype is a blend of the two alleles. Imagine a flower where a red allele and a white allele combine to produce pink petals. In other cases, both alleles are fully expressed simultaneously, a phenomenon known as codominance. Human blood types, AB blood for example, where both A and B alleles are expressed, are a classic illustration of this. And then we delve into the truly intricate tapestry of traits influenced by multiple genes. Most of the characteristics we readily observe in ourselves and others, height, skin color, intelligence, susceptibility to many common diseases, are not determined by a single gene, but by the combined action of many genes, often interacting with environmental factors. This is called polygenic inheritance, and it explains the continuous spectrum of variation we see in populations rather than distinct categories. Furthermore, one gene can sometimes influence the expression of another entirely different gene, a phenomenon called epistasis, adding yet another layer of complexity to the genetic dance. How then are these intricate combinations of alleles passed down through the generations? The answer lies in the remarkable process of sexual reproduction. When an organism produces gametes, sperm or egg cells, a specialized cell division called meiosis occurs. During meiosis, the organism's two sets of chromosomes, one from each parent, are shuffled and then divided, so each gamete receives only one set of chromosomes, a unique combination of alleles. This shuffling is not random. It involves the independent assortment of chromosomes and a process called crossing over, where segments of homologous chromosomes exchange places. These mechanisms ensure that each gamete is genetically unique, carrying a distinct mix of parental alleles. When a sperm fertilizes an egg, the two unique sets of chromosomes combine, restoring the full complement of genetic material in the new organism. This new individual receives a unique blend of alleles from both parents, a genetic lottery drawn anew with each conception. This constant reshuffling and recombination of alleles through sexual reproduction is a primary engine of genetic variation within populations, providing the raw material upon which natural selection can act, driving evolution forward. So, as we reflect upon the silent, ceaseless work of heredity, we come to appreciate that what we inherit are not fully formed traits, but rather the instructions, the alleles, that, in concert with countless others and in dialogue with the environment, will ultimately shape the organism. It is a system of profound elegance and staggering complexity, a testament to the enduring power of information encoded in the very fabric of life. We have begun to unravel the language of life, to understand how instructions are stored and passed. But this genetic script is not static. It is subject to change, sometimes subtly, sometimes dramatically. How do these changes arise, and what are their consequences for the grand narrative of evolution? This is the next frontier of our quiet contemplation. In our last quiet gathering, we journeyed into the intricate dance of heredity, unraveling the double helix and understanding how the language of life, encoded in DNA, is faithfully copied and beautifully shuffled through sexual reproduction. We marveled at the precision of replication and the creative recombination of meiosis, which together ensure that each new generation carries a unique genetic signature, a fresh hand dealt from the ancestral deck. But even the most diligent scribe copying an ancient manuscript can make an occasional slip. And sometimes these slips, these unexpected departures from the original script, are not errors to be corrected, but sparks of an entirely new story waiting to unfold. Today, we invite you to contemplate these profound shifts, these moments when the genetic script itself changes, and how these changes, alongside other powerful, often subtle forces, continually reshape the tapestry of life. We speak of mutation, genetic drift, and gene flow. Three fundamental architects of evolution, often misunderstood, yet utterly essential to the vibrant, ever-changing world we inhabit. Let us begin with the whispered secret at the heart of all novelty: mutation. Imagine, if you will, the vast, intricate instruction manual that is the genome of every living thing. Billions of letters, faithfully copied, generation after generation. Yet within this monumental task, imperfections sometimes arise. A single letter might be swapped for another, like a typo in a word. A whole sentence might be accidentally deleted, or perhaps duplicated, or even inverted. These are mutations, spontaneous, random alterations to the sequence of DNA. They can occur due to errors during DNA replication, the relentless process of copying life's blueprint, or they can be induced by external agents, silent cosmic rays, or certain chemicals that subtly tamper with the molecular bonds. We often carry a misconception that mutations are inherently harmful, always leading to disease or deformity, and indeed, many mutations are deleterious, disrupting vital functions, leading to conditions we recognize as genetic disorders. But pause with us for a moment and consider the full spectrum of their effects. Many mutations are and neutral, occurring in regions of the DNA that do not code for proteins, or resulting in a change to a protein's amino acid sequence that does not alter its function. These silent shifts accumulate over time, serving as molecular clocks, tracing the divergence of species. And then there are the rare, extraordinary mutations that are beneficial. Imagine a slight change in a protein that makes an enzyme work more efficiently, or a modification to a cell surface receptor that offers resistance to a new pathogen. These fortunate accidents, though infrequent, are the ultimate wellspring of all new genetic variation, the raw material upon which natural selection can act. Without mutation, life would be locked in a static, unchanging form. There would be no new traits, no new adaptations, no grand branching tree of life reaching out into myriad forms. Mutation is, in essence, the universe's way of generating new possibilities, a quiet whisper of change that echoes through generations. It is random, undirected, and utterly indifferent to the needs or desires of the organism. Yet, from this randomness, the astonishing diversity of life has sprung. Once these new variations, these alleles born of mutation, exist within a population, their fate is not solely determined by the elegant pressures of natural selection. There are other, more capricious forces at play, particularly when we consider the intimate scale of small populations. Let us turn our contemplative gaze now to genetic drift. Imagine a small, isolated village where, by chance, all the people with blue eyes happen to have more children than those with brown eyes in a particular generation. This isn't because blue eyes offer any advantage. It's simply a matter of luck, of random chance in who survives, who finds a mate, and who successfully reproduces. Over time, In this small village, the frequency of the blue eye allele might increase dramatically, even to the point where brown eyes disappear entirely, purely by chance. This is genetic drift, the random fluctuation of allele frequencies in a population from one generation to the next, a phenomenon most pronounced in small populations. Think of it like drawing marbles from a bag. If you have a large bag with 500 red marbles and 500 blue marbles, and you draw 100, you're likely to get close to 50 red and 50 blue. But if you have a tiny bag with only 5 red and 5 blue marbles, and you draw 2, you might easily pick 2 red ones, drastically changing the proportion for the next round. Genetic drift is a non-adaptive evolutionary force. It does not lead to organisms becoming better suited to their environment. Instead, it can lead to the loss of genetic variation within a population, as certain alleles might randomly disappear, or conversely become fixed, reach 100% frequency, simply due to chance events. We can observe genetic drift playing out in 2 particularly poignant scenarios. Consider the bottleneck effect. Imagine a thriving population of thousands suddenly decimated by a natural disaster, a wildfire, a flood, a prolonged drought. Only a small random handful of individuals survive. The genetic makeup of this surviving remnant is unlikely to be representative of the original larger population. Certain alleles might be overrepresented, others underrepresented, and some might be lost entirely, not because they were disadvantageous, but purely by chance. The future generations will carry the limited genetic diversity of these survivors, even if the population eventually recovers its size. We see echoes of this in endangered species, where past bottlenecks have left them with dangerously low genetic variation, making them vulnerable to disease or environmental changes. Then there is the founder effect. Picture a small group of individuals breaking away from a larger population to establish a new colony elsewhere. Perhaps a few birds are blown off course to a distant island, or a handful of individuals migrate to an isolated valley. This founding group carries only a subset of the alleles present in the original population. The genetic diversity of the new population will therefore be limited to that carried by the founders, and certain alleles rare in the original population might become common in the new one, again purely by chance. This is why isolated human populations or island species often show unique genetic characteristics. So, while natural selection refines and adapts, genetic drift introduces an element of unpredictability, a reminder that not all evolutionary change is about improvement or fitness. Sometimes it's just about who happened to be in the right place at the right time, or who by sheer chance reproduced more. Now let us broaden our perspective beyond the confines of a single population and consider the movement that connects them. Imagine the bustling exchange not just of goods or ideas, but of life's very instructions. This is gene flow. The transfer of alleles from one population to another. Picture a valley where 2 populations of wildflowers bloom on opposite slopes. Occasionally a bee, laden with pollen, might travel from one slope to the other, carrying the genetic material, the alleles, of one population into the gene pool of the other. Or perhaps a few young deer, venturing beyond their birth territory, join a neighbouring herd, introducing new alleles with their arrival. Gene flow is this constant, often subtle, exchange of genetic information. What are its profound effects? Firstly, gene flow tends to increase genetic variation. within a population. If a new allele arises in one population, or if an allele that was lost due to drift is reintroduced, gene flow can bring it into another, enriching its genetic diversity. Secondly, and just as importantly, gene flow tends to decrease genetic differentiation between populations. By constantly mixing alleles, it acts as a homogenizing force, making distant populations more genetically similar to one another. It works against the isolating effects of genetic drift and even against strong local selection pressures, preventing populations from diverging too rapidly. Imagine two populations of fish in adjacent lakes. If a river periodically connects the lakes, allowing fish to move between them, their gene pools will remain relatively similar. But if a landslide permanently separates the lakes, preventing any further gene flow, then genetic drift and local selection pressures will likely cause the two populations to diverge over time, perhaps even leading to speciation. Gene flow, therefore, is a vital thread in the web of life, ensuring a degree of interconnectedness and shared genetic heritage across landscapes and even continents. It reminds us that populations are rarely truly isolated islands. Rather, they are often porous entities, exchanging genetic whispers and shouts that continually reshape their collective identity. As we gently draw this contemplation to a close, consider how these three forces, mutation, genetic drift, and gene flow, interact with natural selection to paint the vibrant, ever-changing portrait of life. Mutation provides the initial brushstrokes, the raw colours and textures, entirely by chance. Genetic drift, particularly in smaller populations, adds an element of unpredictable, non-adaptive shading, sometimes simplifying the palette. Gene flow ensures the colours are not confined to a single canvas, but can spread and mix, creating continuity and preventing stark isolation. And all the while, natural selection acts as the discerning artist, subtly refining, adapting and enhancing the masterpiece, favouring those combinations of traits that best harmonise with the environment. These are not merely abstract concepts. They are the unseen, dynamic forces shaping every organism, every ecosystem, every moment of life's grand narrative. They remind us that the story of evolution is not just one of grand design or inevitable progress, but also one of chance, of movement, and of the profound creativity that springs from imperfection itself. We are, each of us, a testament to this ongoing, magnificent dance of change. As we gently journey through the intricate dance of life's mechanisms, we've traced the subtle currents of change that ripple through generations, the spontaneous whispers of mutation, the random currents of genetic drift, the steady flow of genes between populations, all meticulously sculpted by the selective hand of nature. But what happens when these countless tiny alterations accumulate, not over mere years or centuries, but across the unfathomable expanse of geological time? What grand patterns emerge when these micro-evolutionary forces are given eons to weave their story? To truly grasp the majesty of life's evolution, we must first attune ourselves to a concept that challenges our fleeting human perception. Deep time. Imagine, if you will, that the entire history of our Earth, from its fiery birth to this very moment, is compressed into a single, quiet year. In this cosmic year, the first stirrings of life would appear sometime in March, single-celled organisms dominating the stage for the longest stretch. The first animals would only emerge in late October, dinosaurs would reign supreme for a mere 2 weeks in mid-December, and humanity, our entire recorded history, our civilizations, our dreams, would flash into existence in the final seconds of New Year's Eve. This vast, almost incomprehensible scale is the canvas upon which macroevolution paints its masterpieces. Where microevolution describes the changes within populations and species over shorter timescales, macroevolution refers to the grander narrative, evolutionary change above the species level. It encompasses the origin of new taxonomic groups, the emergence of major evolutionary innovations, and the large-scale patterns of biodiversity and extinction that have shaped our planet over hundreds of millions, even billions of years. It is the story of how a single common ancestor gave rise to the breathtaking diversity we witness today, from the simplest bacterium to the towering redwood, from the deepest sea anglerfish to you, contemplating these words. To visualize this profound interconnectedness, we often speak of the tree of life. It is not a ladder, with humanity proudly perched at the top, but rather a sprawling, endlessly branching arboreal metaphor. Every living organism, extant or extinct, represents a twig on this magnificent tree, and every fork in a branch signifies a speciation event, a moment when one lineage diverged into two. The deeper you trace a branch, the further back in time you travel, until all branches converge at a single ancient root, the last universal common ancestor, or Luca, from which all life on Earth is believed to have descended. Consider the implications of this image. It means that you share a common ancestor, not just with the chimpanzee, but with the oak tree outside your window, the mushroom pushing through the forest floor, and the bacteria swirling in a drop of pond water. The molecular machinery that drives your cells, the very language of DNA, the elegant dance of protein synthesis, echoes the fundamental processes found in every other living thing. We build these intricate phylogenetic trees by comparing observable traits, yes, but primarily by analyzing the molecular blueprints themselves, DNA and protein sequences. The more similar the sequences, the more recently two species shared a common ancestor. It is a testament to the elegant parsimony of life that such a vast array of forms can arise from variations on a shared ancient theme. Throughout this immense span of deep time, life has undergone truly transformative events, moments of profound innovation that reshaped the entire biosphere. Imagine, for instance, the quiet revolution of endosymbiosis. Billions of years ago, we believed that an ancient host cell engulfed but did not digest, a free-living bacterium capable of aerobic respiration. Rather than becoming a meal, this bacterium found a safe haven, and, in return, provided energy for its host. Over eons, this symbiotic relationship deepened, the bacterium evolving into what we now recognize as the mitochondrion, the powerhouses of nearly all eukaryotic cells, including your own. A similar event involving a photosynthetic bacterium gave rise to chloroplasts in plant cells. These were not mere steps, they were leaps that redefined the very architecture of life. Later, around 540 million years ago, the Earth witnessed the Cambrian explosion, a relatively sudden burst of diversification that saw the rapid emergence of most major animal body plans we recognize today. Before this period, life was predominantly soft-bodied and simple. Then, almost as if a dam broke, a proliferation of hard shells, complex eyes, and specialized appendages appeared in the fossil record. What triggered this explosion of novelty remains a subject of intense scientific inquiry. Perhaps rising oxygen levels, new predator-prey dynamics, or the evolution of developmental genes that allowed for greater morphological complexity, and then the monumental challenge of colonizing land. Imagine the ancient oceans teeming with life, while the continents lay barren, exposed to harsh UV radiation and desiccation. Yet, over hundreds of millions of years, plants evolved cuticles to prevent water loss, roots to anchor and absorb, and vascular tissues to transport water and nutrients. Animals followed, developing lungs, limbs, and protective skins, slowly, painstakingly adapting to a world fundamentally different from the one they left behind. Each of these transitions was not a single event, but a protracted series of adaptations, driven by the same micro-evolutionary forces we've discussed, accumulating through countless generations. But the story of life is not solely one of diversification and innovation. It is also profoundly shaped by loss. Extinction is a natural, ongoing process, the inevitable consequence of a dynamic world where environments change and species fail to adapt. The vast majority of species that have ever lived are now extinct, their branches pruned from the tree of life. Yet occasionally, the Earth has experienced cataclysmic events that trigger mass extinctions. Periods where a significant percentage of all species vanish in a geologically short time. The most famous, perhaps, is the Cretaceous-Paleogene (KPG) extinction event, approximately 66 million years ago, which saw the demise of the non-avian dinosaurs, likely triggered by a massive asteroid impact. While devastating, these events are not merely destructive, they are also creative forces. By clearing ecological niches, mass extinctions open vast opportunities for the surviving lineages to diversify and radiate, fundamentally reshaping the trajectory of life. The rise of mammals, for instance, into the dominant large terrestrial animals was direct consequence of the dinosaurs' disappearance. As we contemplate these grand narratives, it is crucial to gently set aside some persistent misconceptions about macroevolution. The notion of missing links, for instance, implies a linear progression, a gap in a chain. Instead, the fossil record, though incomplete, presents us with a continuous spectrum of transitional forms. Organisms that exhibit a mosaic of traits from ancestral and descendant groups. Archaeopteryx, with its feathers and reptilian skeleton, is not a missing link between reptiles and birds, but a beautiful example of a transitional fossil, a testament to the gradual branching nature of evolution. Similarly, macroevolution is not a march towards perfection or a predetermined goal. It is a blind, opportunistic process, driven by the contingencies of environment and the random variations arising within populations. There is no pinnacle of evolution, no ultimate design. Every species alive today, including our own, is simply a lineage that has successfully navigated the challenges of existence up to this moment. A temporary triumph in an ongoing, ever-shifting saga. We too are but a twig on this magnificent ancient tree. Our very existence, our consciousness, our capacity for wonder, are products of billions of years of cosmic and biological evolution. To understand macroevolution is to understand our deep kinship with all life, to feel the echoes of ancient oceans in our blood, the legacy of photosynthesis in the air we breathe, and the shared molecular heritage that binds us to every creature, great and small. It is a profound lesson in humility and interconnectedness, urging us to recognize our place not as masters of this planet, but as an integral yet fragile part of its ongoing majestic story. With this vast perspective in mind, we might begin to ask, if life is so interconnected and species are constantly evolving, what then defines the very concept of life itself? And how do we distinguish it from the non-living world, especially as we consider its origins and its potential future? The line, we shall find, is often more blurred, more dynamic, and far more fascinating than we might initially imagine.